welcome everybody to a presentation by the Department of Psychiatry at the School of Clinical Sciences here at MONASH University. My name is Suresh Sundram. I'm the head of the Department of Psychiatry, and we've got a number of scientists joining us today who'll be presenting, as well as Brendan Gillespie, who's a current PhD student with us, who'd be very happy and keen to answer any questions or queries that people might have about being a PhD student in the department. Brendan's a first year student. So if people have questions as we go along, please feel free to type them in the chat box and we'll answer them or endeavour to answer them at the end of the presentations. The Department of Psychiatry, as you may know, is one of the major clinical areas in medicine and is one of the core subjects in fourth year medicine, it being a prerequisite for graduating or training as a doctor. And then obviously a significant number of people choose to do psychiatry as a specialty, a clinical specialty. It is, as you might suspect, a fairly major area of clinical research. And we also here at the School of Clinical Sciences have a very active basic research program. The way that we'll structure this evening's presentation is that we'll talk about two of the major streams of research within the department. One of them is a very clinically facing area of research in refugee and asylum seeker mental health, and the other is in the translational molecular psychiatry program, which is aiming towards a precision psychiatry. And I'll describe that a little bit more in detail when we get to that section. So Rachel, if I could ask you just to move to the next slide. So the people who'll be talking today, I'll be presenting on the asylum seeker and refugee mental health aspect of the program, and then I'll introduce the precision psychiatry area before handing over to Dr. Rachel Hill and Dr. Joohyung Lee, who are two of the senior researchers in that laboratory, or those laboratories, plural, to talk more about that area of work. So Rachel, if I get you to move to the next slide. This is about the asylum seeker and refugee mental health research program. It is, as I mentioned before, a very clinically oriented program, so I'm not sure how it would fit in with people who are listening to this particular session in terms of their interest. As people will know at the moment, or may know at the moment, the current situation with regards to refugees and asylum seekers is as problematic as it has ever been in human history, with more than 70 million people who are displaced, both internally displaced as well as refugees in other countries, many millions of whom are seeking refuge in third countries where they may be able to gain protection. Unfortunately, over half of these people are children, and that presents really a major public health, if not a global health risk going forward. Next slide, please, Rachel. The area of health when it comes to refugees and asylum seekers that we're most interested in, obviously, is their mental health. And the way that we divide up mental health of refugees and asylum seekers is to think about health in three different phases. We think about it in terms of the pre-migration trauma that drove the individual or the family to leave their community, to leave their society, to leave their place of residence to seek refuge elsewhere. We think about their migration journey and their transit environment, how it is that they got from their home to another country, both as a transit country in search of a permanent place of refuge, or that permanent place of refuge, be that Australia or other countries that accept refugees. And then we think about the post-migration environment. That is, what is the environment that the person or the family now finds themselves in, and how does that environment contribute to their recovery, contribute to their sense of well-being, or potentially contribute to their ongoing deterioration with reference to their mental health. Next slide, please, Rachel. The way that we think about this is that the asylum seekers and refugees who find themselves in Australia, and in particular in Melbourne, have a whole host of experiences from a psychological perspective. And these have not been well characterised. They've been described in varying contexts with regards to refugees who've gone to settle in lots of different countries, especially in Western industrialised nations. However, the plight of the asylum seeker specifically has not been well understood. And the research program that we've undertaken here at Monash University is about trying to better understand and characterise the psychological characteristics of people who are seeking asylum, seeking refuge in a country such as Australia. And I've put some of those descriptors of their experience and of the types of psychological responses that they undergo on that slide. Next slide, please, Rachel. So what it is that we're trying to further investigate, and these are all areas that prospective students might wish to engage in projects on, is about trying to understand both the prevalence and severity of the types of psychiatric disorder that people exhibit and the factors which contribute towards that psychological and psychiatric disorder. Why it is that people who've had an experience of immigration detention are worse off than people who haven't had such experiences. What are the characteristics that differentiate the experience? What are the qualities that exacerbate or perpetuate psychiatric disorder in detained cohorts? And in particular, at the moment, we're very interested in the concept known as moral injury, which is an area that's receiving a lot of attention, which really describes the experience of somebody who has had an injury to their sense of moral or ethical frame of reference. We're very interested in how these illnesses and disorders progress over time. That is the trajectory of the psychological state. The next point. And what it is about seeking asylum in Australia that might contribute to or exacerbate psychiatric disorder in this cohort. So all of these are current areas of research activity, and we are looking to further our understanding. So if people are interested, more than welcome to come and talk to me, send me an email to discuss areas of interest that might pertain to these areas of work. The next area, we might go into the next slide. The next area is something which is completely different in the sense that what we're trying to do here is to introduce into psychiatry the same area of work and research which has been able to be introduced into other areas of medicine. So in particular in areas such as oncology, which is cancer medicine, immunology, autoimmune disorders, gastroenterology. It is about trying to adopt a precision medicine approach to psychiatry. And of course, the challenge that we have in psychiatry is that the only way that we can think about psychiatric disorder, the only way that we can identify and diagnose people is on the basis of their symptoms, the symptoms that they describe to us, and the clinical features that we might observe in a face-to-face interaction with the individual. And what we've got here on this slide are some of the symptom domains that we see in schizophrenia. However, the challenge is that these symptoms are neither specific nor are they selective for this particular disorder. So if we go to the next slide and bring up the whole slide. Yeah, thank you. No, yeah, that's right, thank you. The description here is in fact that the symptoms that people describe with schizophrenia pertain to a whole range of psychiatric disorders, including things such as major depression, things such as bipolar disorder, and disorders such as autism, for example. And that's what you can see in the bottom there on the left-hand side. What we have, however, is this complete admixture of symptoms and clinical features across a range of disorders. And we understand that there are a whole set of biological factors and processes which contribute towards the presentation of these disorders in any one individual. So we've got some of these described here as genetic risk factors, molecules which may be identified as being abnormal, cells which have been identified as being potentially altered, as well as circuits and behaviors that we can observe in individuals. However, basing the description or the segregation of these patients on what we can observe, that is the behavior and the self-report of patients, is not in any way going to allow us to understand what it is that are the genetic basis for these disorders, nor do we understand what are the molecules that might be dysregulated or disrupted as a consequence of those genetic changes. And as a consequence of that, we can't ever really fully pass or separate patients out into the biological underpinnings or the biological basis of the pathologies which cause these variations or these various disorders. So the task that we're faced with is to try and find ways of classifying these disorders based on their biological basis, which will allow us then to be able to develop targeted interventions which will treat and potentially modify the course of these disorders. I hesitate to use the word cure, but ultimately what we would like to do is to define pathological subtypes which we understand the biological basis of, for which we can develop specific and targeted treatments. And that's what we can see there on the right-hand side at the bottom of the slide. So the next slide, Rachel, and if we put those elements up. So what we're doing in the Translational Molecular Psychiatry program is to see whether we can use a translational pipeline approach to understanding these disorders. And the way that we do that is to try and identify clusters of individuals, in particular through family pedigrees, who are at risk. We then seek to collect a very detailed clinical understanding of those patients in conjunction with whole exome or whole genome sequencing of those individuals to identify genetic risk factors. Those genetic risk factors we probe for in two ways. The first way is using post-mortem human brain, so that is post-mortem human brain tissue from people who have had the disorder and healthy controls, where we use a range of genetic, molecular, and cellular approaches to understanding what might be disrupted or dysregulated in the postmortem human brain tissue, and then by also modeling those genetic risk factors in animal models. And I'll allow Rachel and Ju to subsequently describe that in more detail. But by modeling those particular genetic risk factors in animal models, what we hope to be able to do is to understand the cellular pathways and the molecules which are dysregulated, with the view that we can then ultimately design drugs and therapeutic tests which will target those specific genes in a way that can be corrective and modify the disease. And then, of course, the final and most important part of this translational pipeline is to trial these new agents back in cohorts of patients to be able to demonstrate that these particular treatments or interventions are going to be effective. And it is only in having that iterative loop, if you like, that iterative pathway, that we can modify the disease on the basis of understanding it from a molecular and genetic perspective. So obviously in that slide that I've got there in front of you, there are multiple opportunities to undertake PhDs in all those different areas, right from the clinical perspectives through the genetic and molecular perspectives, through to post-mortem human brain, as well as then animal and molecular and drug design perspectives. So I'll stop there and hand over to Rachel, who will be able to talk about a number of these elements in much greater detail.
Okay, thanks, Suresh. So my lab are the Behavioural Neuroscience Laboratory, and we are part of this Translational Psychiatry Research Program. And our focus is really on this aspect here, as Suresh mentioned, the preclinical side of things. Okay. So what we are trying to do is model risk factors associated with schizophrenia, and these include both genetic but also environmental risk factors. We model them in mice. We look at a number of different behaviours in the mice to see how these risk factors affect specific behavioural domains, and we also probe the molecular biology of these mice, so cellular and molecular biology, and with the hopes that we can identify new targets for therapeutic development to bring back to the clinic. So at the moment, in terms of modelling risk factors associated with schizophrenia, we actually are currently modelling an environmental risk factor, which is maternal immune activation. So we know from the literature, there's a lot of data, epidemiological data, that suggests that if a mother gets an infection when she's pregnant, it significantly increases the risk of her offspring to develop schizophrenia later in life. And we want to understand how that happens, and then how can we therefore intervene before this happens. And this is particularly relevant at the moment with this significant COVID-19 pandemic. You know, what are the effects of this pandemic on pregnant women and their developing babies and their brain development? So we model this in mice. We get pregnant dams and expose them to a maternal infection. And then we look at the offspring later in life. And we also look sort of immediately after the infection to try to understand how this is having this effect. We've also got a few genetically modified mice. This mouse here is an NMDA receptor knockout mouse. And we know that glutamate is very much, glutamate synthesis and neurotransmitter activation is very much altered in schizophrenia. And so we're studying this mouse at the moment. We've also got a beta cellular knockout mouse. And this is a very exciting mouse, which stemmed from Suresh's work in the clinic, where he identified that the beta cellular molecule is reduced in people with schizophrenia. And we want to understand what that molecule does in the brain, and why not having it or having lower levels can increase your risk of getting schizophrenia. So we've got a knockout mouse, and we're looking at the behaviour and the neurobiology of this mouse at the moment. So we look at the behaviour in a number of different ways. We can also do recordings of the firing of neurons within the brain while the mouse is doing a behaviour. So we can really look at sort of the neurobiological underpinnings of what's happening while the mouse is performing different tasks. And this is just a graphical representation of different neural firing patterns while the mouse is in this maze here. We've also got these fantastic new touchscreen apparatus, which was designed based on the sort of cognitive tasks that we do in humans, so that we can directly translate our mouse findings to what happens in humans. For this task here, the mouse is one of the lights here on this sort of like a mini iPad shines up, and the mouse is trained to poke the light. It has to come back here and cross a beam, an infrared beam. And then when it's presented, when it turns around again, it's presented with two lights, and it has to press with its nose the light that it didn't press before. So it's a non-matching to location task that's quite often done with humans. And we train our mice to do this, and if they get it right, they get a little strawberry milkshake reward, which is situated in the back here. So if you have a look at this mouse, I'm going to play the video, and it's actually very good at doing this task. So you can see it presses the light, comes back and crosses the beam, and then it presses the non-matching light, and then it rushes back to get its strawberry milkshake reward. So we found that in our immune activation model, the offspring here are not able to perform this task to a similar degree as what our controls are. And we're also looking at this task in our knockout models as well. We can also do some cellular labelling, and this is a new technique that we've just established in our lab, where we can map different dendrites stemming off the neurons, and we can do all sorts of morphological measurements on these dendrites in terms of their size, their spine density, etc. And we're finding out more and more every day from these sort of images and this sort of analysis. So if you're interested in doing research in our group, we've got a number of different models, and you can do anything from behaviour and electrophysiology to cellular and molecular experiments. And if you are further interested and want to read more about what we do, these are a few recent publications that we've had. And this is my email address here. And then I think if no one's got any imminent questions, we'll move on to Joo's presentation.
Yeah, next slide, Rachel. Thank you. Hi. Oh, slides look a little bit different. Anyways, I'm Joo. So my group's interested in, I guess, two research topics mainly. So the first topic revolves around Parkinson's disease. So as you may be aware, it's a very common neurodegenerative disorder, and it's caused by, I guess, loss of these dopamine-containing neurons in the midbrain. And what that leads to is the eventual, I guess, loss in voluntary control of movement. So the main, I guess, unmet need in Parkinson's disease is how do we stop the progression, or how do we even slow the progression of this dopamine cell loss? So the target has been mainly focused around, so our interest lies in this particular gene called SRY, which is a gene that's found on the Y chromosome, so it's only in males. And we've got some exciting, I guess, preliminary or published evidence showing that SRY may be a disease-modifying target. So that this may actually provide explanation why males are more vulnerable to Parkinson's disease, and thereby if you target this gene, you might be able to actually slow or stop the disease progression in male Parkinson's disease patients, which is a highly, I guess, innovative approach. Another aspect of Parkinson's disease we are interested in is this surgical procedure known as the deep brain stimulation, which is essentially an electrode that is implanted into the basal ganglia. So it's part that's involved in the symptom evolution in Parkinson's disease. And what it does is basically to correct the abnormal brain rhythm in Parkinson's disease. So it's actually a symptomatic therapy, but what we are starting to understand is that there might actually be additional effects. It might actually be trophic or neuroprotective effects. So we're trying to understand whether there could actually be a novel procedure or a target to actually slow, prevent or stop Parkinson's disease. Another aspect of Parkinson's disease we're interested in is the consequence of long-term dopamine therapy. So there's a drug called levodopa. It's been the hallmark treatment for Parkinson's disease, but after about 5 years of L-dopa treatment, what you get is the evolution or the genesis of these dyskinesias. So it's a side effect that's resulting from this abnormal dopamine receptor stimulation. So we found, again, this particular target known as receptor activity modifying protein, or RAMS, that could be also a novel way to symptomatically reduce L-dopa-induced dyskinesia. So thereby you could actually still take the medication, but not have these disabling, I guess, side effects. So the second part of our research revolves around the concept of brain-sex differences. So we want to better understand really the biological basis of why male and female's brains differ. And potentially that may provide an explanation why males and females are more vulnerable to certain types of disorders. So females, for example, are much more prone to getting Alzheimer's disease or affective disorders like depression or anxiety. On the other hand, males are more prevalent or males are more vulnerable to disorders like ADHD, autism, as well as Parkinson's disease. So a lot of the effort, I guess, over the last 20, 30 years has focused on understanding how the sex hormones actually, affect the sex differences. And there's been a lot of great studies, including stuff, earlier work from Rachel and also current work, I guess, from Rachel. what I'm interested in, I guess, is this emerging field of sex chromosomes, in particular, as I mentioned, this gene called SRY. So we want to better understand where SRY is actually expressed in the male brain by using our unique antibodies that we've generated in-house to identify type of cells where SRY is expressed, and then better understanding the function of SRY by using pharmacological approach to knock down SRY expression, and then looking at the behavioral consequences on such knockdown. So what we've shown in earlier or prelim work is that there is certainly a role for SRY in these dopamine neurons and in relation to voluntary movement, and consequently Parkinson's disease, which I discussed a little bit earlier. And then better understanding, I guess, the role of SRY in the normal brain might actually give us some answer or a novel sort of insight into why males are more vulnerable to these disorders like autism and ADHD, which you see the striking sex difference somewhere between 4:1 or even 11:1 for severe forms of ASD, where males are predominantly affected. So we might actually provide a novel genetic explanation for these male-biased neurodevelopmental disorders. Next slide, Rach. Thank you. So again, my group revolves around using a multidisciplinary approach. So we use a variety of cell culture models. We use a lot of immortalized cell lines from which are neuronal in origin or microglial in origin. And what we try to use the in vitro model is to really interrogate the signaling mechanisms underlying these drug targets. And also serves as a very useful tool for high throughput drug screening through a lot of assays that involve around oxidative stress or mitochondrial dysfunction, cell viability, and so forth. And then we can also, in parallel, test the effect of modulating our drug targets using pharmacological approaches, as well as using a variety of genetic and toxin-based animal models of Parkinson's disease, where we can look at the symptoms by destroying or selectively causing the loss of these dopamine neurons, and looking at the effect of these drugs to see whether we can actually slow or halt the disease progression. And we can see that behavior through a number of motor behavioral assays we've established in the lab. And then at the end of the behavioral studies, we look at the brains to see whether we've actually rescued these dopamine neurons as well as striatal dopamine terminals, and then also look at whether some of the signaling pathways that are known to be dysregulated are also affected. And finally, we combined, I guess, the work from the in vitro and in vivo models with human post-mortem brains to really better understand whether these drug targets are clinically relevant. So looking at, I guess, post-mortem brains taken from Parkinson's disease patients, for example, we then can identify what type of cells our drug targets are expressed and potentially dysregulated, and when is this dysregulation occurring during the disease process. So again, we have a multitude of projects that are revolving around, I guess, largely around Parkinson's disease, but certainly this new area of brain sex differences. And feel free to give me an email at the PowerPoint slide in the first, but I think that will be the end of my presentation. Thank you.
Great, thanks. Thanks, Zhu. So just to conclude, and please feel free to type questions in the chat box if you'd like to. You can see from the presentations by Rachel and by Zhu, what we're really trying to understand is what is the neuroscientific, neurobiological basis for a range of disorders which previously have really only been present as disorders of the mind. And it's about trying to relate them back to how the brain is disturbed, and as a consequence of understanding what is the neurobiological basis for these disruptions in brain function, to be able to develop new treatments for these disorders. I think it's important to state two things here. The first is that what we're really doing is working at the very cutting edge of neuroscience, and what we can push the limits of our understanding of the human brain and how it relates to all those intangible characteristics such as behavior, emotions, thoughts. And secondly, it's about trying to improve the lives of people who are profoundly affected by some of the most severe and debilitating disorders known to mankind, which are the psychiatric and neuropsychiatric disorders. And I think it's for those two reasons that so many of us are profoundly and deeply attracted, are drawn to this area of research and endeavour. And I think it's an area that in the next decade or next 2 decades, we'll see probably the greatest advances in medical research that there are to have. So we think it's pretty exciting. We'd be very keen to hear from you. And if there are any questions that people have, please feel free to ask them now, either by typing in the box is probably the easiest way. I don't know, Brendan, whether you're going to actually say anything. So I can hand over the screen to you if you like.
Yeah, sure. So I've known Rachel and Suresh for a couple of years, and I've always found this sort of research to be very, very multifaceted. It's not very normal that you can find a group that, you know, they look at psychology, physiology, neuroscience, and can bring all those concepts together. So that's one of the reasons that I really, really enjoyed this sort of research myself, and I found them to be a great team to work with. And if you have any questions, you're more than welcome to contact me as well.
Okay, my screen has frozen, so I can see that there's a question there, but I can't actually open it. Is anybody else open it?
Yeah, so I can read it. So Kiara's just asking if they need to complete an APAC accredited course to focus on research in psychiatry. So I thought this might be a better one for you, Suresh. Might be more across those.
It could be, but I'm not clear what an APAC course is. Maybe Rachel might be able to understand that.
Yeah, I'm not clear what an APAC accredited course is either.
Not.
I thought maybe you would know that, Suresh, but
I can answer that. An APAC accredited course is an undergraduate course that is accredited with to become a psychologist. So the undergraduate psychology courses at Monash are APAC accredited, and you have to have that to become a psychologist. So whether that's a prerequisite to do this, which probably isn't, because people can come from science courses, is that correct?
Yeah, so we had a question that was similar to that just before, but you don't need to do any APAC accredited courses.
Looks like Suresh's screen might have frozen.
Yeah, so the answer is basically no, you don't need to have done an APAC accredited course. We have a whole neuroscience program that all comes through from biomedical sciences and from the Department of Anatomy. So yeah, just a science degree is fine.
I think that's the only question we have there in the Q&A.
Okay.
We have answered a couple throughout the session. Does anybody else have any other questions? No, I don't think so. So Rachel, do you just want to wrap it up and let people know how to contact potential supervisors, and then I think we can end the session.
Sure. So thanks everyone for attending today. I hope you've enjoyed the presentation and you're as excited by this research as we are. We're all very passionate researchers, and we really do feel that with this sort of pipeline of translational research, we can actually start to make a difference in the lives of people living with these severe psychiatric and neurological disorders. If you'd like to contact us, I believe our email addresses are on there, but I can also direct you to, there's a link to some research pages that I believe Vithia was going to be sending around.
Yes, she's popped them in the chat, so you can see them in the chat there as well.
Okay, great. And we're accepting honours and PhD students at this point in time.
Great, thanks Rachel.
Okay.
Thanks everybody.